Today we continue the class lectures on industrial bioprocessing. The topic for today is production kinetics. The objective is to investigate production kinetics and the limiting factors. Mainly, it aims to characterize product formation based on yield. When we talk about product formation rate, kinetics means rate of formation of the product. We would like to know the relationship between product synthesis and energy generations in the cell. So there are three types of the growth productions kinetics as listed here, whether it is growth associated or non-growth associated. Growth means cell growth. Mixed mode means a combination of two of these. For example, if you recall the primary metabolite which are produced during the exponential phase or growth phase, so these are called products produced together with the self-growth. That's why it's called growth associated. This usually involve those enzymes which is produced together with the cell. For example, glucose isomerase. Also other metabolite intermediates such as pyruvate, citrates, acetate, and so on. As for the non-growth, meaning that this is already entering into the stationary phase of the self-growth for the production of secondary metabolites such as antibiotic and so on. Then the third type of kinetic is mixed growth associated, which meaning that the product can be formed during the exponential phase as well as the stationary phase. So for example, lactate, ethanol, and also other secondary metabolite. So these three diagrams can give you a better idea. For example, growth associated, when the cell grows from lag phase to exponential phase, from the beginning also the product is already formed inside the cell biomass, for example E. coli and so on, continuously from the beginning until for the entire growth phase. So this is called growth associated product kinetics. If the product is only produced during the stationary phase, this is called non-growth associated. But for anything that happen between exponential and stationary, this is called mixed mode, mixed growth associated. One of the common bioproducts is called monoclonal antibody, which is produced using mammalian cells such as hybridomas. This also can involve growth associated as well as non-growth associated. Mainly since 1990, there was a Japanese professor who found this negatively growth associated product formation kinetic. This is closely related to non-growth, almost going into negatively growth, meaning that the growth rate of the cell is very low, so that the production of the antibody can be more long-lasting. For example, we can add some inhibitor to inhibit the production of protein. Then the cell will go into a kind of very slow growth rate. If the growth rate can be slow by 50%, it was reported that the antibody can be enhanced by 130%. Okay. Another way of slowing down the cell growth is adding this rRNA inhibitor, for example, actinomycin D, a kind of antibiotic. So this equation shows the formation rate of product. dP over dt means that the change of product form over a specific duration of time. RP, rate of reaction. If you have taken some subject of related to chemical reaction engineering, so you should know it is represented by this common short form R. R means rate of reactions. P denote product. Okay, is expressed as a function of X, which is biomass concentration. QP is the self-growth, not self-growth, sorry, product formation constant, QP. So this is a generalized production equation. And since product formation is usually related to energy metabolism, as I told you in the previous lectures that when microbial digest substrate, for example glucose, it will generate ATP while breaking down the substrate to a smaller nutrient for assimilation. So this is related to cellular energy demand, because the cell growth process itself is an energy intensive process where product will be formed whenever there is a growth. Okay, look at this equation. RP, product formation rate, is as a function of X, self biomass concentration. This is meaning to produce more cells of, it can be E. coli, yeast, and so on. All of this rate constant, they must have a constant here. So QP is the product formation rate constant. Rx is, X means a cell, okay? For the rate of formation of cell biomass is as a function of mu, the growth rate, exponential growth rate of the cell. Also, we can write it as a dx over dt, how many cells is being formed over a certain duration of time. But importantly, as I mentioned, for this whole PowerPoint, we concerned about the yield, how much of the product, how many more of product can be formed over how many more of cell biomass was being formed. Okay, so this is called yield for product over biomass. We can calculate by dividing RP over Rx. So quite easy to remember. Any Y is the rate of the P formation over the rate of the biomass form. P, here P, X, X. Okay. So if we put these two equations together, cancel off the X, you can get QP over mu. However, this equation is oversimplification of what exactly will happen. Later on, you will see why. This is because when we talk about product formation, right, substrate consumption is not just used to produce the product alone. From the metabolism, ATP, the energy produced, will be used, a part of producing the biomass. It also will be used for the maintenance of cell, because the cell will need to move around for motility. It will need to duplicate more. That's why it's called turnover of cellular components. And also we have to adjust, energy is needed to adjust the membrane potentials for nutrient transport across the membrane cell, and also to adjust the internal pH. This YPX is only the theoretical yield. In reality, when we do experiment, whatever we observe and we calculate is somehow there is another value called Y dash P over X, and this is the observed one, and the value is higher than the theoretical yield. So we have two yield for product formation over biomass. form. From the experimental work, we can calculate this one, where when we divided Rp over Rx, again, P over X here, we put Rp, this equation that we have derived just now, again, we divided to with mu x, yeah, x cancel off, we rearrange, it will give this equation, yeah. So this is something that we can observe, we can measure from the experiment. And if we know the MP from another experiment, we can find the if we know the observed YP and also the mu, we can find YPX theoretical and also MP from the experimental linear equation. So okay, recap again, this is the dx over dt, okay, how the cell growth, x is concentration for the biomass, going through exponential to stationary and then to the death phase. This process will be limited by the depletions of resources such as nutrients, space, and oxygen, okay. Just now, whatever equation that we have derived were mainly on the cell growth and product formation coupled with energy metabolism, yeah. So we can relate from the ATP consumption, but there are also some other processes in which the product formation is indirectly coupled with energy metabolism. So this usually will involve more complex function of the growth rate and growth rate constant, which will be derived from the experiment. So this is just for your information, comparing different type of product from this one, yeah, from here, relate RS and RP. RS and RP, yeah, so this term is for product formation, and then the first term X, you know, this term is telling you the substrate, the overall rate of substrate consumption is depending on how fast the rate that is used, the substrate is used to generate the X, and how they will result in the yield of the biomass over how many substrate being consumed, plus how many substrate is being used to form the product over how many yield of product over the substrate, and then of course how many substrate is used to maintain the cell. So when we rearrange this, we put all the X outside here and tidy up the equation. This entire term is called QS, yeah, because the consumption rate of the substrate is as a function of rate constant for substrate and X here, okay. So this QS has three terms, yeah. Remember, growth, to support the growth of the cell. Secondly, to support the formation of the product, and thirdly, for maintenance, yeah, okay. And then Rx, recall Rx, the growth curve, yeah, the formation of biomass is as a function of growth rate, mu x, yeah. So the mu here, that's why the Rx become mu x here. So if we look at this complex equation, there are so many parameters, 1, 2, 3, 4, 5, that we need to determine from the experimental in order to capture how fast the consumption rate of the substrate, yeah. Why this is so important? Later on I will clarify. Coming back to yield of cell from substrate, yeah. So this is important. As I mentioned just now, the apparent or the observed yield for how many cell that's biomass that are formed over substrate is governed by Rx over Rs, X, S, yeah? So you can see X over S here, mu X, QS, X, yeah? So the QS, remember the QS, cancel off the X, we have mu and QS. So these three terms represent QS, yeah? As I mentioned already. cell growth, to support cell growth, substrate to support product formation, substrate to support maintenance. Okay, so this is observed step by step. Firstly, we assume QP equal to 0, then find MS. Previous slide is X over S. Okay? But now we want to know P over S. How do we derive the yield? Always remember rate of P formation over rate of S being consumed. Yeah. So this equation, if you do not remember, you can go back to slide 13 again. And RS, as you remember, QS X. Yeah, QS got 3 terms. Again, remember this substrate used to support microbial cell growth, substrate used to support product formation, substrate used for maintenance. Yeah, so all these 3 terms represent QS. So cancel off the X. In other words, we may not have to do more experiment to find MP and YPX. Yeah, we can rely on this equation to sort out. Okay, but I know it is quite complicated. Later on, I believe I give you the 5 tutorial with already the answer sheet. All these are the theoretical background on how those equations are derived and how to solve the tutorial question. So I hope you have tried out the tutorial, and with this theoretical background, you can better understand what is RS, RP, R, and also the yields related to substrate, related to product formation and biomass formation. Okay, so this is a summary that we need in order to develop the yield. We need to conduct experiment to get the data for calculating the substrate uptake rate RS with and without product formation. Yeah, you can refer to slide 18 if you do not remember. And then similarly, we also wanted to know how is the pattern, the rate for the product generation with time. We call it RP. You can refer slide 6 and 13 and maybe more. Also with and without cell growth. Yeah, so meaning to say some of the product can be formed during stationary or exponential phase. But in order to get the data for all these, at the same time, we also have to monitor how the cell grow over the time. Then only we know whether the product is formed during the exponential or stationary phase. Okay? And we will know when the product start to form, whether it is during exponential or stationary. So all these 3 are interrelated, and typically we just have to run the experiment along the fermentation. We monitor how the cell is being growth. Yeah, for every 1 hour we will take the sample and we will read through optical density using UV spectrophotometer within the wavelength of 450 to 600. Then we can know from the optical density, we can know how many biomass is being produced. So this X is the biomass concentration. So ultimately, we wanted to calculate what is the yield. Of course, because we do the fermentation, we have invested millions of dollars to buy the fermenter, buy the culture, buy the medium, construct all the utility for sterilization. We want to harvest the product. Yeah, of course, we want to calculate the yield. And before we really do the experiment, conducted the large-scale fermentation, as a biochemical engineer, we have to estimate how many nutritions which is required. On last lecture, we have done some stoichiometry on how to calculate for any specific stoichiometric equation, how many mole of substrate is needed to produce certain product. Yeah, so that is also closely related to yield calculations. But from the stoichiometric, we cannot know the rate. So that's why we have to study the kinetics in this lecture to estimate RS. Okay, and from the growth curve also, understanding the relationship of X biomass growth pattern and the product formation rate at different stage of the cell growth rate, we can manipulate how is the relationship of substrate concentration with X formation or P formation, so that we can maximize our yield of P over substrate. Okay, so MS and MP are the maintenance term. These are the factors to explain why the YPS theoretical is greater than the observed. I think this should be the other way around, right? I think the dash is the larger one. This one needs some revisit later on. Let me continue first. In order to, our ultimate goal of this chapter is to maximize the production of our product. Okay, so we refresh again this equation. During the formation of product, there is the concern on how to grow the cell and also maintain non-growth. This is MP is the maintenance constant for product formation. So how do we control? How do we alter all these parameters? From the last few lectures, we stress on the control of the nutrient supply. It doesn't mean you supply a lot of nutrient, it will give you a lot of product. It's not necessarily like this. If you sometimes product formation can be inhibited by high substrate concentration. Okay, or if the nutrient is too high, it will suddenly produce too many biomass cell and the product formation is low. Okay, so there is a good, that's why we sometimes prefer fed batch feeding mode to feed the nutrients slowly to match the maximum product formation rate. And also there is the oxygen supply we have to control, and typically we like fed batch process. And if we select those microbial strains that can produce higher product, this we can enhance our yield. Okay, this is typically done through recombinant DNA technique. For example, you can put in more promoter to actively express the protein of the desired product. This can be done through metabolic engineering. Okay, so there is this paper entitled Growth, Metabolic and Antibody Production Kinetics of Hybridoma Cell Culture. It elaborate the effect of serum concentration. This is the substrate, and then the control of the dissolved oxygen and the control of pH in the batch reactor to maximize the productions of antibody, monoclonal antibody. So there are some notes here, I believe you can read. So this is to tell you how to adjust. For example, dissolved oxygen can control the formation of ATP. So if the too much oxygen, if you have too much ATP form, this is saying that oxidative phosphorylation accounted for about 60% of total energy production. So under certain suboptimal pH or dissolved oxygen content, it will produce too much ATP that will go to this oxidative, the metabolism of the glucose happening too fast through the oxidative phosphorylation. So sometimes you may have to lower the DO or lower the serum concentration in order to avoid too much ATP go to the oxidation. So we want to make sure the growth rate, the cell growth rate to be lower. This is why going into the stage of not growth associated by controlling some of these parameters. But this is saying that, however, the antibody production rate is not dependent on the serum and DO, yeah, okay, but somehow it is depending on the pH. When the pH is get below 7.2, the product antibody production rate can be increased by twofold. Okay, so a lot of the different observation can be obtained by adjusting this major parameter. So they are also controlling of the substrate nutrient content. Okay, and monitoring the amino acid metabolism can also check whether the serum level, whether there is an interrelationship or not. Okay, so this paper I encourage you to read. Okay, and then for example, you may be given some case study like this, yeah, so you should be able to interpret, yeah, the impact of substrate concentration on product formation. Okay, so you can see that when we alter the initial substrate concentration, yeah, so this is a substrate concentration at a specific time. Okay, if it is a batch, then each of these dot represent an individual separate experiment, yeah? But this can also refer to fed batch, where the substrate concentration can be increased over certain time. So regardless of the time, at any time when the substrate concentration is so, it tells you that, yeah, when the mu, the growth rate is lower at 0.1, cell growth rate, the mu, at a slower growth cell growth rate, yeah, when you increase the substrate concentration, the product formation is quite poor, yeah, as compared to when the cell is growth at a higher rate, 0.2, increasing the substrate concentration will significantly enhance the product formation. So in this case, we have to manipulate the fermentation to try to increase the cell growth rate to a higher level at 0.2 in order to enhance the product formation. Okay, so this is we will do experiment by controlling the substrate. Yeah, by controlling the substrate, we can achieve we can control different growth rate of the cell, either at 0.1 or 0.2, and then at different substrate concentration, you can see different P formation. Yeah? So this is plotted into the graph like this. Okay. Another way of interpreting that graph, yeah, you can see that when the substrate increases, product is being formed. Yeah, so in the exam, if you are given a graph like this, you should be able to explain, okay, when at the low yield of the biomass over substrate, yeah, lower yield, okay, meaning that when the biomass cell is generated at a lower rate, yeah, this blue color, it promote higher product formation. This is what I have told you already. This is generally is like this, yeah, because why? Most of the substrate is used to produce the product instead of forming the cell. Yeah, so you can imagine if a lot of the substrate, the nutrient is used to form the cell, yeah, the higher value of the yield of the cell, then you have lower product formation. Okay, so this is quite typical trend. Yeah, so I hope today's course will give you a clearer idea on how do we relate product formation rate to and substrate uptake rate to cell growth rate, and how we we know how to derive various yield, yeah, based on those rates. And with this theoretical explanation, I hope you will be able to work out the tutorial questions in a clearer manners. Yeah, okay, thank you.